Hello everyone, I am Aranjyoti. I hope you all are fine. Today we will continue the second chapter of class 8, Microorganisms, Friend and Foe. Now let's proceed to the mind map of this chapter. This is the mind map of the chapter. We already learned in this chapter microorganisms, type of microorganisms, useful microbes like they used in preparation of bread, formation of curd, clean the environment, formation of alcohol and increase the fertility of the soil. Then we learned about harmful microbes. They cause the disease in animals, plants and humans. Now we will discuss food preservation and nitrogen cycle in this chapter. Now let's proceed to the chapter. Now, first term of today's topic is food preservation. What is a food preservation? Food preservation is just a technique in which we can be used to prevent the food from spoiling. What is a food preservation? It's just a technique in which we can prevent the food from spoiling. For this technique, we can be use different methods like freezing, heating, pasteurization and certain chemicals. We can be used to preserve the food from the microorganisms. There is a one question, how do we preserve cooked food at home? We can be preserved cooked food at home by keeping the food in refrigerator and reheating the food. In this way, we will preserve the cooked food. After that, some microorganisms also spoil our food. When the foods are spoiled, that food emit bad smell, bad taste and color of food is also changed. When the food is spoiled, which type of food is there? That is, it emit bad smell, bad taste and change in the color of the food also. Now, spoiling of food is a chemical reaction. Yes, spoiling of food is a chemical reaction. It involves the breakdown of the food's particle by the microbes. It goes through acidic reaction and breakdown of the food into simpler form. This way we can explain spoiling of food is a chemical reaction. Now we can be discuss how we can be preserve our food in homes. For this we can be have certain methods. First method we can be used to preserve the food at home. That method is known as a chemical method. In this method we can use salt and edible oil for chemicals generally used to check the growth of the microorganisms, and salt and edibles generally known as the preservatives. What is a preservatives? Preservatives is a substance or a chemical that is added to food product to prevent decomposition by microbial growths or by undesirable chemical changes. Same way, we add salt and acid preservative to pickle to prevent the attack of microbes. There are the common preservatives, that is sodium benzoate and sodium metabisulfate. These are the chemicals preservative we can be used to preserve our food, and they also used in the jams and sketches also to check the spoilage. Now next method for food preservation is common salt. This method we generally use to check the growth of bacteria for meat and fish. How we can be check? Meat and fish we generally covered with dry salt. This way we can be check the growth of the bacteria. And salting we can also use to preserve amla, raw mango and tamarind. Next method is preservation by sugar. How sugar is used to prevent the food for spoiling? Sugar reduce the moisture contact which inhibits the growth of bacteria which spoil food. Means sugar reduce the moisture in the food, and that moisture that we can be removed, they prevent the growth of the bacteria and our food is safe. Next method is preservation by oil and vinegar. These are the two more preservative we can be used for food preservation. Oil and vinegar generally prevent spoilage of pickle, because generally these two ingredients we can be used in formation of pickles, because bacteria cannot live in such environment where we use oil and vinegar both. Vegetables, fruit, fish, meat are often preserved by this method. I hope everyone understand the methods which we are using to preserve the food. Our next method is known as a heat and cold treatment. These two method we generally doing in our homes. Heat, like we are reheating the food to preserve the microbes from the food, and reheating means we generally kill the harmful microorganisms that will spoil the food. And second method is heat cooling. So for cooling we can be keep our food in the refrigerator. Why we keep our food in refrigerator? Because in the refrigerator the temperature is low as compared to our surrounding. So low temperature also inhibits the growth of microorganisms. Everyone understand how we can preserve the food by the method of heat and cold treatment. Now, why does the milk that come in packet are not spoiled? The answer of this question is that milk is pasteurized milk. How we can be pasteurize the milk? For this technique, we can be heat the milk at about 70 degree Celsius. And when we heat up the milk at this temperature, the harmful microbes they generally kill, and we heat the milk approximately 15 to 30 seconds and then suddenly chilled and stored. By this method we can prevent the growth of microbes, and this method is known as a pasteurization. What is the pasteurization? To heating the milk about 70 degrees Celsius for 15 to 30 seconds and then suddenly chilled and stored. This method is known as a pasteurization. This method or process is discovered by Louis Pasteur. Our next method is storage and packing. Storage is also very important method to preserve the food. If we will not store our food properly, then the food get spoiled. How it will be get spoiled? Because if the moisture is in the food, so that moisture will germinate the microbes and that microbes will spoil the food. So proper storage should be there while preserving the food items. Now next is packing. These days dry fruits and even vegetables are sold in the sealed airtight packets. Why they used airtight packets? They use the airtight packets. The moisture will not go inside the packet. If the moisture is there, then the attack of microbes should be there. So that's why we can be use the sealed airtight packet to prevent our foods from the microbes. These are the certain method we can be used to preserve our food from the microorganisms. Now next topic is nitrogen fixation. Nitrogen fixation is the process in which we can be fix the atmospheric nitrogen in soil. How we can be fix these nitrogen in soil? By the help of bacteria that is known as a rhizobium. Rhizobium generally found in the roots of the leguminous plants. Leguminous plants such as bean, peas and pulses. And they have a symbiotic relationship. What is the meaning of symbiotic relationship? The two different organisms live together, share their shelter and nutrients. Sometimes nitrogen gets fixed through the action of lightning, but how, but you know that the amount of nitrogen in the atmosphere remain constant. Then how the nitrogen remain fixed in the atmosphere by using these bacteria we can be discussed in the nitrogen cycle. Students, we already learn in the amount of nitrogen in our atmosphere, that is 78%, and this is very important constituents for protein, chlorophyll, nucleic acid and vitamin. The atmospheric nitrogen is fixed by the two methods. First method is nitrogen fixed by a bacteria, that is blue green algae, and second method is lightning. Firstly, we will discuss about how the blue green algae fix the nitrogen from the atmosphere. Firstly, they will take the nitrogen from the atmosphere. Who will take? Blue green algae. They will take the nitrogen from the atmosphere. Then there are the certain bacteria, they fix nitrogen into nitrogenous compound, and these nitrogenous compound, that is, goes into the soil. When these nitrogenous compound fix in the soil, then the leguminous plant, they will take the nitrogen from the soil with the help of roots, and in the roots they have a one bacteria, rhizobium. They will take the nitrogen from the soil and the plant will take the nitrogen. After that, the animal eat the plants. When the animals die and they waste their excretion, they again mix in the soil. After that, again the soil will get the compounds of nitrogen. Then further, there are some bacteria, they turn the compound of nitrogen into gaseous form, and this gaseous form of nitrogen is also go in the atmosphere. In this way, the amount of nitrogen in atmosphere remain fixed. Now next we will be discuss about how lightning is help. First of all, lightning is a huge amount of electrical sparks, and these electrical sparks help to break down the strong bonds of the nitrogen and convert it into nitrogenous compound, and this nitrogenous compound is in the soil. After that, when this nitrogenous compound is in the soil, there are the certain bacteria, they convert the nitrogenous compound into gaseous form, and this gaseous form of nitrogen again goes to the atmosphere. In these two ways, you can be understand how the amount of nitrogen is fixed in the atmosphere, and this cycle is also known as a nitrogen cycle. I hope everyone understand this cycle. This cycle is very important. By this cycle we finish the second chapter. Now let's see which things we already learned in this chapter. We learned types of bacteria, useful microbes, harmful microbes, food preservation, different methods we used for food preservation. At last, we already discussed nitrogen cycle. How the nitrogen cycle will work? It will work on the two methods. First is by blue-green algae and second one is by lighting. This way, the amount of nitrogen remain fixed in the atmosphere. So everyone write the question whatever I explained in this video on your notebooks. I hope everyone understand this chapter. Thank you and have a nice day.